Hi everyone, thanks for joining us. Today we're going to be presenting some of our work on reference collections in microarchaeology using experimental archaeology. This presentation came out of our current work and conversation on deductive reference collections, which we're hoping will culminate in a paper that will be published this year in an open access journal. This is an example of the type of dialogue and discussion we want to encourage in building reference collections for microscopy work. Throughout this presentation, we want to show that how we create these reference collections is as important as the ways that we use these reference collections in our analysis. But first, we will provide a quick overview of what reference collections are and how we use them in archaeology.
We would like to start this presentation with a phrase from Edmund Lockhart, who is considered by the majority of scholars as the father of forensic science. Every contact leaves a trace. Lockhart was probably the first scientist to fully recognize the value of trace evidence and formulated the principle of exchange. With contact between two items, there will be an exchange, and effectively is that trace that we try to interpret in the archaeological record from objects, soil, and many other lines of evidence. Many archaeologists, therefore, that are working on trace analysis will recognize their work in that of Lockhart. Next. So a brief overview on historical collection. In general, we can say that reference collection started in very ancient times with the role of collectors, and some are really famous, like Herodotus and Pliny, who went around, travel a lot, collecting objects and both from the natural world and made by people to document variety of environment and human behavior. Reference collection became also very important from the late 1700s, I would say, when travelers around the world, when scientists, botanists, zoologists traveled around the world a lot and basically brought back example of what they were seeing. And this is an important recording of life in geological form of unknown lands. Today, these are valuable archives of no longer existing form of life. For example, some animal that became extinct as consequence of human from Victorian time and with development of forensic science, the reference collection began to play an important role from fighting crimes to food safety. And in archaeology, they help us to decipher the past. But what are effectively reference collection? How do they work? Reference collection are example of traces, both from object on object and from the environment around. And their principle is very simple. They work by comparison. These comparisons are the strengths and the weakness of the reference collections. Effectively, because it's the role of the analyst in this process is crucial. Here you have an example of ancient starch in the bottom picture on the left and a modern reference material that contains starch of barley above. The reference collection need, however, to reflect complex realities, and this paper will show you a little bit through case study, will give you a taste of what the complexity of this collection are, and the collection need also to be large.
So the first example that I wanted to introduce is from the site of Starcar, which is a Mesolithic site in Yorkshire. And this is a site that Jess and I have been working on for some time now. And the area that we're focusing in for the purposes of our reference collection is the creation of beads, in this case primarily from shale and from amber, using flint tools. We think probably awls and possibly a number of mèches de forêt, a specialist type of awl. And we're interested in the array of macroscopic traces of modification that we can see in these tools: snap tips, rounded edges, and some of the initial micro-wear trace work that's been done previously and published in 2018 that looks like we can see wear traces on these tools as well. So we're looking to expand this and to look at the range of usages for these awls and try and get a better handle on how these tools wear in relation to the production of this type of assemblage of beads. So this has started a dialogue between Jess, who is an expert with microwear analysis and microscopy, and myself, who has a background here with experimental work, and I've made beads of this type over a number of years and taught students to do so and so on. And so we have a lot to bring to this dialogue as we try to understand how different methods of trying to create beads with this type of stone tool might leave different macroscopic traces that I could see during my experimental work, and Jess might see differently with the microware traces that are developed. And this allows us to move back and forth as we expand and develop our reference collection. And this workflow Jess will describe.
Yeah, so as you can see on the slide, there is a really key back and forth dialogue between the experimental and the microware analysis. And so this started off initially with perforating a single bead, so in this instance, shale. And then from that, so Andy would perforate a single bead, and then he would give me the Mèche de Fourré tool to and analyze under the microscope. And as you can see from this, there were no discernible wear traces from a single bead. So then we increased the number of beads that Andy was perforating, stopping at regular intervals to check the microwear development. And this back and forth dialogue was applied to a range of different quantities of beads in different materials, as will be discussed later. And having this sort of analysis as we went along with the back and forth between the microwear and the experimental meant that we didn't create this hierarchy in the techniques with the separation between the experimenter and the analyst, which is often the case with reference collections. By working together, we had a chance to discuss the working processes. So, for example, as Andy mentioned, when macroscopic observations were noted by him during the production process, I was able to then see how that translated on the polish development under the microscope, which meant that we were able to observe and track the development and the life process of each tool that was used and made in the reference collection. This dialogue also helped us to understand some taphonomic traces observed on the archaeological tools. So, for example, working shale is unlikely to cause tip breakage. And as an analyst, I had the closest interaction with the archaeological tool. And then I was able to feed back that information that I was observing to Andy, who was then able to explore the different techniques and working processes that may have caused what I was seeing under the microscope on the archaeological tools. This enabled us to observe some of the complexities in the archaeological record, and the feedback from myself allowed Andy to think more dynamically about the production processes, because although the experimenter may have certain processes and production methods in mind when they're looking at how beads were made, the archaeological record doesn't always follow a functional approach. And so this allowed us to be more actualistic and realistic in the development of our reference collection.
So we can kind of explore that theme as we've rolled out our experiments to other materials that would have been manipulated at Star-Kar during the Mesolithic thousands of years ago. So as we start to look at bone, at wood, at perforating hide, possibly linked to, say, clothing production, we can start to think about the types of microwear traces that Jess identifies and feed this into the likely and less likely methods of creation or use of these different materials with ulls in this case. So it allows me as the experimenter to better steer the experiment and adapt the experiment depending on whether we're getting closer or further away to the types of microwear traces that Jess is identifying. And I can be sensitive and feed back in the types of macro modifications that I'm seeing in the tool. So let's say we work with a harder material that necessarily is more abrasive and destructive to the tool, and I can feed that in to allow us to contextualize the type and extent of wear that we might be seeing. And Jess can demonstrate this through shale and how wear develops as the tool and the life of the tool expands out as I use the tool more.
Yeah, so as you can see on the slides here, we've got a micrograph, so that's an image taken under the microscope of a tool that was used to make 20 shale beads, so a sort of a smaller amount of beads for a smaller amount of time, and then all the way through to an awl that was used to create 60 shale beads. And this is exactly the type of reference collection we were able to create and document for each different material that we used and experimented with. These reference collections not only help us to identify and interpret the objects used by past individuals, as we see with the case study at Star-Kar, but they also can help us to explore the byproducts created by individuals and the environments around them, which Anita will talk about now.
Thank you. So reference collection for the identification of micro-remains are built because micro-remains from plants and animals are found in a variety of archaeological contexts like soil, pottery, artifacts, and even mineralized dental plaque, which is known as calculus. These tiny microscopic fragments of plants and animals can inform us about a great variety of aspects of people and animal diet, the environment they live in, and health in ancient times. And to give you an idea of the importance of this reference collection to be as wide as possible and the type of reasoning you need to have behind, I want to show you this example from my PhD. The remains in the red square, picture 1 and 2, are very similar by the look of it. However, picture 1, you have tiny fragments of a moth or a butterfly wing, the scale of the animal. And in picture 2, you have something that sometimes is found in a plant cell, and they are called calcium oxalate drugs. And is basically a deposit. As you can see at glance, they can look really similar. When in the archaeological record from calculus, from my medieval sample from Leicester, I retrieved this variety of debris that you see in the black squares from picture 3 to 8. You can see that it can be quite tricky at glance to tell which one they are. Calcium oxalate from plants, which is picture 2, dissolve in hydrochloric acid. So it was very easy for me to understand if these were plants or animals. But if you don't take that approach of wide environment around, so the people may have eaten plant, but they also were in contact in an environment where remains of insects were, you wouldn't be able to tell the difference probably. Then next, please. In this case, I want to give you also the example of corn cockle from my own experimental work. Corn cockle is a wild plant and also a weed of crops. It became almost extinct in the past 200 years because we changed the way we farm crop, but it is a plant that has been found in archaeological contests, sometimes in high quantity, since the beginning of agriculture, and also the Neolithic period, basically. And in high quantity, this plant is toxic if ingested in quantity. So if you find it, it can be an indication of potential food hygiene issue. Now, what it was interesting for me is that when I reviewed the experimental work of others, for example, in crops and starch analysis, I noticed that the weeds were almost never included in the experimental work. So when I prepare my reference material to see micro debris from crops from the Middle Ages, how they would look under the microscope, I made sure that together with the crop, like wheat, barley, I included the remains of weeds and seed of weeds that would be common at the time in which the crop were grown in the Middle Ages. And here it is, the archaeological evidence that that approach was successful. This on the left, you have in red, picture 1 show you how the tiny fragment of the external part of the seed of corn cockle, shown in picture 2, look like under the microscope. And in picture 4, 5, and 6, you see example of these fragments found from dental calculus of a medieval individual, which meant that effectively people, this individual at least, and many others in the same population that had this remain, had eaten a crop that was in which these weeds this corn cockle weed was present. If I had not taken an experimental archaeology approach that widened to a better reality, more close to what people were eating in the past, I would have probably never been able to identify this plant.
So we hope from these case studies, we've been able to highlight the importance of actualistic and realistic experiments when we're building and creating reference collections. The actualism seen through the ecological diversity and the need for a reference collection to reflect the range of species that would have been found in the archaeological record, and actualism in how we define accuracy. So if tool A and tool B were used for the same amount of time, that's not particularly reflective of human action. We need to measure the time taken for actual tasks rather than superfluous timings taken to work particular materials. We really hope that our discussion has presented some ideas about the ways we can start a conversation about reference collections and how they're created and implemented in our analyses. We would really like to hear about your own approaches to reference collections too, whether we need perhaps more detailed ways of recording and documenting reference collections, or maybe sharing our collections to encourage future collaborative work with access to a range of different materials from different sources. Thanks so much for listening. If you would like to have a look at updates and news about our paper on deductive reference collections, please do check out the Year Centre social media accounts where we will be posting any updates. Thanks again, and we look forward to hearing your comments.